Hello, we're going to work on some Punnett Square practice today. And so we'll start off by doing your bell ringer. So that's what is marked here first in your Google Classroom. And our bell ringer is looking at crossing a brown-eyed dog with a blue-eyed dog. So we're going to cross a brown-eyed dog and a blue-eyed dog. When we do that, we're going to use the information that we know about these dogs, and that is that we've got chromosomes for each of them. And we're going to say that this is chromosome 4 for the dogs. So on chromosome 4, there's going to be a place on that chromosome where there is a gene that codes for eye color. So for the brown-eyed dog, that's going to be, for our purposes, homozygous dominant. And for the blue-eyed dog, it's going to be homozygous recessive. And so there's the little eye color place for the genes. Okay, and that's just those little marks across there are just showing where the eye color genes are located on the chromosomes. So what does that mean in terms of using the capital B's or the lowercase b's? That was a question that we got several times today. And really, the uppercase B's and lowercase b's, we just pick a letter. The letter doesn't matter. What does matter is whether we use uppercase or lowercase. And I'm going to scroll down just a little bit here, and I'm going to show you two sequences of DNA. And if you look at these two sequences of DNA, you should be able to see that they're different. So they're just a little bit different here and here. And those differences are important for us because that tells us that we have two different codes. This codes for a protein, and it's all kind of wadded up like that, that will give us brown eyes. This sequence codes for a protein that will give us blue eyes. So those proteins have slightly different structures, and they result in different colors of eyes. So the irises of the dogs are going to be a little bit different. They're going to reflect or absorb different colors of light. That's essentially what happens. So there are a couple of ways that you can figure out which of these is dominant, which is recessive. And what we know through both sequencing, the sequencing gives us the actual sequence here of the DNA, and through breeding dogs for generations, is that if a dog has two copies of this code, then it will definitely have brown eyes. We also know that if the dog has one copy of this code and one copy of this code, then the dog will have brown eyes also. Only if we end up with two copies of this code will we end up with a dog with blue eyes. So that tells us that this code over here that I'm going to mark in pink, this is the dominant form of the gene for eye color, and I'm going to mark this in orange. This is the recessive form, recessive. This is not a very sensitive screen here, so the writing doesn't come out very good. So when we have these two things, when we have the dominant identified and the recessive identified, then we can decide how we want to denote them, how we want to identify them. And so we'll use the capital letter for the dominant, and so that would be a capital B, and we use the lowercase letter for the recessive, and so that would be a lowercase b. So instead of having to write out the whole code, and the code would probably be about 1,000 bases, we've only got like 5 here, so that'd be a lot more bases to write out. We can just substitute and put in a capital letter or a lowercase letter, and that lets us know what the code is, whether it's the dominant code or the recessive code that's found on the chromosomes. So when we make our crosses, then when we breed these organisms, we can figure out what we're going to see, simply because we've used different letters to represent that. So we know that from what we've done so far, we know that we can have a chromosome in each of the gametes that are produced. We know that we will have a chromosome in each of the gametes that are produced from each dog. So the brown-eyed dog will have a chromosome in each gamete. So it would have that the one that I just underlined in yellow, in one gamete. This is going to be in another gamete. You can have other copies of these in the other two gametes. So when we write this out, we would have this form and this form separately that could be passed on. And we know that the same thing happens with the blue-eyed dog. So you could have this form and this form passed on. So when these combine, you get a chromosome from each parent. And when you get that chromosome from each parent, in green, you have the offspring. And this is showing that we have some genetic variation here. Although the phenotypes are going to be similar, they're all going to be brown, so they're going to be similar to one of the parents, the actual genotype, so the letters that we see, are different from either parent. So this is giving us some indication that there's some genetic variation. This also is showing us exactly how the cross occurred. Now, I put this in yesterday's video, but just in case you didn't watch that, or you need to see it again for today, at the end of Tuesday, we worked on making sure that Zoe and Grover, making sure of what kind of offspring they could produce if they were crossed. So Zoe and Grover both have the same genotypes. It's heterozygous for red lips, and that means that they have both a dominant and a recessive allele. So both a capital letter and a lowercase letter would be showing up here. And so this shows you what the possible offspring would be from a cross between Zoe and Grover. So check what you have. Make sure that you have this from yesterday. And then you are going to work on the back part here. If you haven't already done that, you are going to choose two other crosses. You're going to choose two other crosses to perform right here. And so you're going to pick two characters. And that shows you right there. You're going to pick two characters. And if you go back to the Sesame Street Genetics page 2, you can see what the genotypes are for all of the characters. So you can just go back over there and see what you need to pick. Pick your trait. So are you going to use lips? Are you going to use whether it has hair or not? What are you going to use? And then you're going to identify the genotypes of your characters. That's in number 3. So what are their genotypes? You're going to actually do the cross in at this point for the Punnett square. And then you're going to look at the offspring. And remember that the offspring will be right here in those 4 cells. So what are the genotypes? Genotypes mean what types of the genes are present. Phenotypes, you're looking at the physical characteristics. So you're going to write out what the physical characteristics are. These are going to be different for different groups based on who you chose to cross and what trait you're choosing to cross. Once you finish cross 1, go ahead to cross 2, and then there's a think about it type of question down at the end. this covers the majority of what we did in class today. There were maybe 2 classes that got through part of the slides on number 3. I'm not going to cover that today. I'm going to cover that tomorrow so that I can cover all of it at one time. So you don't really have to worry about, if you're doing makeup, you don't have to worry about number 3 here on your work for today. Make sure that you check your class. I'm just looking at one of the classes here for this video, but make sure that you check your class to see specifically whether you have homework tonight and to see how far we got in this work for today. Thanks and have a lovely day.